me show you the actual experiment from the original paper right here. What Hugh Pelham's lab did was he took BIP and PDI, as both of you were suggesting. The reason why, as many of you were suggesting, the reason why you might choose BIP and PDI is they're completely different proteins. BIP is doing folding, PDI is making disulfide bonds. So their active sites are going, the sequences that are responsible for those activities are going to be completely different from each other, right? Because they're different activities. So when you compare the sequences, those are not going to line up. But taking very disparate proteins, you might find the common sequence, right? So that I think was pretty brilliant to compare BIP and PDI, and many of you suggested that. And then what Hugh Pelham's lab also did was said, this, you know, the secretory pathway is conserved across phylogeny. So I'm not only going to compare BIP and PDI, I'm going to compare BIP and PDI from vertebrates, insects, plants, fungi, and protozoa, looking for the common sequence that's in all of those. And then the noise will sort of fall away. And when they did that exact experiment, which is an experiment, even though you're not getting your hands wet yet, what they found was that the very last four, there was some variation, but if you looked at the very end of the protein at the C terminus, the signal could have been anywhere, but it turned out at the very C terminus, the last 4 amino acids were well, the last one was always leucine, so the C-terminal one was always leucine. And then if we go 4 from the end, it tended to be lysine, then K, D, E, L. So that's the 3-letter code. There was more variation depending on the species on the 4th one, not within a species, but like HDEL is the sequence that's found in some yeast, and then occasionally RGEL. But most commonly, and always for mammalian cells, it is KDEL. And so the sequence is KDEL, and it's at the very C terminus. Both BIP and PDI have that sequence. And then they could test whether that sequence is necessary for BIP and PDI being retained in the ER. But before we look at that experiment, which you all came up with, let's go to another Instapol question. You construct a plasmid that expresses a mutant fibronectin protein. That's a soluble protein that has KDEL added to the C terminus. Where will be its final location? 2, 1. And the vast majority of you said that if you take fibronectin and add KDEL to the C terminus, it would be found in the ER. And none of you came up with that experiment. You came up with a variation of that. This is a different kind of experiment. So once the sequence has been identified on secretory proteins, on ER resident proteins, you could mutate it and see whether it remains in the ER, right? That's it. This is a variation. It's taking that signal and adding it to a protein that would usually not be retained in the ER. So you're asking, and we'll have more to say about this kind of testing of signals later, but you're asking, is this signal sufficient for retaining the ER, the protein in the ER? And you all recognize that that the mutant fibronectin would be found in the ER. And that's the experiment that Hugh Pelham did, and I'm going to show you the result from that paper. This was before GFP, so this is looking at a cell. You can't see the entire cell, but you're looking at a secreted protein that would be like fibronectin, and they expressed a mutant form of it, so that had KDEL added to it, and you get this distribution with the wild type. You get this distribution with the lysosomal, with this lysozymal protein that would usually be secreted here. And what you're looking at here, you can't see the rest of the cell, but you're looking at the slowest step along the secretory pathway. You end up, it's secreted. You can't see that because it's all washed away once it's secreted. But this is the distribution you'd expect for a secreted protein. Boom, completely different with KDEL added to the C terminus. Now we've got the ER distribution, this ER thing. this ER network. And that's showing that KDEL is sufficient for targeting. It's all you have to add. If we look at the signal and we see, maybe you would be fooled and thinking EL is the signal. But if you added that to, that's part of the signal, it's necessary for the signal, but it's not sufficient. If you took a secretory protein and added EL to the C-terminus, you would get this distribution. It's not the entire signal. So we'll have more to say about this, but these are tests to show that a signal is necessary, what's necessary for it, and what's sufficient. Questions? Okay, so how does KDEL signal, how does the signal target proteins back to the ER? So we identified the signal, but we don't yet know what are the molecular proteins that are responsible for that, for bringing it back to the ER. And the accessory proteins, and some of these were identified with yeast work with yeast. And how accessory proteins, how KDEL, the signal works is, do we need a break? We're good. Still pretty alert? Okay, we'll do a little break a little bit later. How the KDEL signal targets proteins back to the ER is using another protein that was identified with this genetic screen called the KDEL receptor, which is a receptor for KDEL-containing proteins. And this is how it works. So we have here, it's shown in, this is meant to show the ER right here. We have a COP2 coated vesicle budding off, and it contains not only secretory proteins shown in blue, but also soluble resident proteins like BIP and PDI, because those are just scooped up. And those are delivered to the cis Golgi. The secretory proteins then will continue up through the Golgi and secreted. But the KDEL-containing proteins like BIP and PDI are returned in COP1 coated vesicles. And they do so because they have KDEL at their C-terminus, and they bind to a receptor right here called the KDEL receptor that spans the membrane, so it's going to be stuck in the bilayer, and it binds to these KDEL-containing proteins and is targeted to COP1 coated vesicles, these vesicles that are responsible for retrograde transport. These have the appropriate V snares and V snares, so they'll recognize the T snares on the endoplasmic reticulum. The coat falls off, the V snares and T snares bind, and this is delivered back to the endoplasmic reticulum. What targets the KDEL receptor to these COP1 coated vesicles is another signal. So the cytoplasmic tail of the KDEL receptor has a signal that is pretty loose. It's 2 lysines with particular amino acids after them, so it's called KKXX, and it's somewhere in this cytoplasmic tail. And what this signal does is it will bind to COP1 coat proteins, and that's how they're concentrated into COP1 coated vesicles to come along. And then the KDEL containing proteins are bound and dragged along for the ride. Questions? Yeah?
So does that mean that the KDEL receptor also binds to those proteins in the ER?
Oh, really good question. So I love your question. So you have here KDEL containing, the KDEL receptor, you're picturing right here, it's not even drawn here, but when this vesicle fuses right here, you're going to have KDEL containing proteins right here, right? KDEL receptors in this ER. So then it would be a futile cycle unless there was a way for the KDEL receptor to bind proteins in the cis Golgi and release them and fail to bind KDEL containing proteins in the ER. Otherwise, it would be continuously cycling like that, right? So you're recognizing the conundrum that exists, and we're about to address that. Other questions? Okay, keep that question in mind. Yeah?
I still don't understand how BIP and PDI don't get like smoothed up in like the Golgi like they do, like in the ER, and like how they get.
Okay, so your question is a further clarification for how BIB and PDI are trafficked back into COP1 coated vesicles instead of continuing along the ER. That's your question, right? Okay, and the reason that they are not, or with very low frequency, continuing along here, is that this receptor, the KDEL receptor, is hanging out in the cis Golgi, and it has a binding affinity in its luminal domain for KDEL. It's actually a shape. So it will bind to KDEL tight enough that then when that receptor goes into COP1 coated vesicles, it will drag along KDEL containing proteins because they are bound to that receptor. So the receptor is here, it's targeted to COP1 coated vesicles, and so KDEL, that is sort of vacuuming up or binding all of the KDEL containing proteins and returning them back to the ER. Does that make sense? Other questions? Or clarifications? Okay, we still haven't explained how the KDEL receptor will bind in the cis Golgi but release in the ER, and that has to happen. But before we answer that question, let's go to another instaful question. You express a mutant actin protein, which is a soluble cytoplasmic protein. We'll have more to say about actin, that has the sequence KDEL added to the carboxy terminus. Where will be its final location? And for some reason, that's not coming out. But it will come out. This is what I love it when I see the correct answer emerging. So originally, we had a bunch of people who picked this right here in black. The KDEL containing, that actin would be found in the endoplasmic reticulum. And then we had a shift to, which one? The mutant actin would be found, here it is, in the cytosol right here. So can somebody who changed their mind, who originally put A, explain why they switched? Yeah, go ahead.
Well, originally I saw KDEL and I was like, oh, so it'll go to the ER, but then I thought about it and I was like, if it's a soluble cytosolic protein, there's not KDEL receptors there. So it'll just say, even though it has that signal receptor.
Right. Yeah, that's right. And I set you up because that's what the first question was doing. I was setting you up, and then we're talking about KDEL, and you're obsessed with KDEL, and then, but once you hear the right answer, you're like, whoa, there's more. It has to see the receptor. And actin is here. It will never experience the KDEL receptor. A protein has to get into the ER in the first place. So all BIP and PDI and all the ER soluble proteins have to begin with an ERSS. As you said, actin is here. It doesn't have an ERSS, so it's going to be here. If you added KDEL to it, it would still be in the cytosol. Perfect thinking. Other questions on that? And I'm going to show you a movie of the KDEL receptor labeled with GFP. So this is a cell right here. This is the Golgi that you're looking at right here. And this cell has there's a trick you can do. It's a temperature sensitive trick where you can, in this particular mutant, you can sort of stop everything at the Golgi and then go to the permissive temperature and then watch things release from the Golgi. And the KDEL receptor has been labeled with GFP and stuck in the Golgi. And then what we're looking at right here is the KDEL receptor actually going from the Golgi back to the ER. And you can see that you know that these are COP1 coated vesicles that are coming off here. You can also see the same thing we saw before, that some of these seem to go on sort of tracks, and the round vesicles are sort of stretched out along tracks. They've lost their COP1 coats, and that bilayer is stretched along these tracks, which we'll see later are microtubules, as we talk more about microtubules. And you all know that these KDEL receptors inside the vesicle are are bound to BIP and PDI, and bringing those soluble proteins back to the ER, as well as the COP1 vesicles having the bilayer that needs to return to the ER. Okay, so how then?